Okay. I did the introduction portion of the experiment along with the research question and hypothesis. We studied the effects of Artemia salina under different concentrations of caffeine. And though caffeine may not seem like a big pollutant issue, it still happens, and we wanted to see how the experiment would affect the growing and hatching rate. Studies have been done before on other species of fish and caffeine, but not as much with brine shrimp. We got help from a study by Collier in 2017 and found the effects that caffeine has on zebrafish and how it could affect us as humans. In our experiment, we tested very, very low concentrations and small amount of brine shrimp. We had to be very precise with the levels or our experiment would fail. Most of our levels like salt, caffeine, water, etc. were from the National Library of Medicine. The research led us to the question of what effects do different concentrations have on the hatching rate of A. salina? We assume that the higher the concentration, the less trace of A. salina would be present. This is because the toxicity levels that caffeine produces and because it's not a natural habitat for the species. Even with lower concentrations, it still can have scarce effects on the ecosystem. Some other questions that we had along the way on our experiment was how much caffeine pollutant is out there? What can the long-term effects of caffeine have on an ecosystem? Without our confounding factors, how different would our experiment have gone? And that's my portion.
Okay. So I did the materials and methods section of our poster, and so we had 3 treatment groups total. And for our experiment, we wanted to make sure they were the same all across. We had 4 containers in each of our treatment groups, making 12 total. And we made sure that they had the same salinity, pH, and that the only change would be the caffeine that we would add to each of the treatments, basically. All of them also had 0.7 grams of A. salina eggs. So with our 3 treatment groups, one was the control, which we just kept salt water and 200 milliliters of water in each cup and just gently mixed them in. And then for our treatment 1, we made sure to add 0.02 grams of caffeine and then gently mix that in with the salt and the water to make sure that it was gently blended, and then they added all the eggs. And then the 3rd treatment was the same 200 milliliter cup of water and with 0.10 grams of caffeine added to the experiment. And then we let them sit for 48 hours before we checked on them and counted how many lived through the experiment. And so for our statistical analysis, we chose one-way ANOVA because we have 3 treatment groups total, and we needed to tell how many survived from each treatment group. So that's what we did. That's my section.
All right, for the results section, we had a total of 3 treatments and 4 trials, which gave us a total of 12 data variables to work with. As you can kind of see with the graph, our experiment indicated a dose-dependent decrease between the increase in caffeine concentration and a decrease in Artemia salina eclosion. So essentially just we're seeing as caffeine increases, we're seeing a somewhat linear relationship in the amount of hatchings decreasing. For treatment 1, we ended with a mean of 43.5 Artemia salina per 2 milliliter sample. In treatment 2, we had a mean of 31.5. Treatment 3 with 0.10 grams was 11.75. So we can see with these means that it is gradually decreasing with a higher caffeine concentration. Our standard deviation, our control group had a standard deviation of 6.24. Our treatment 2 with 0.2 grams of caffeine had a standard deviation of 10.91, which was our highest among the 3 treatment groups. And then treatment 3 had a standard deviation of 2.5. Based on this data, we performed a one-way ANOVA data analysis, and that revealed the p-value of 0.0006, which is relatively low. And that kind of represents that there is a statistical significance between our treatment groups, and this likely is our data wasn't likely just based off of pure chance. And yeah, with that, we'll move on to the discussion section.
So I did the discussion and the future research. For the discussion section, we saw a clear difference between the groups, so we rejected the new hypothesis. As caffeine increased, fewer artemies salina hatched. This shows that caffeine has a negative effect. The control group had the highest number, and the highest caffeine group had the lowest. We also had some problems in the experiment, like lighting, airflow, and not very precise measurements, which could have affected the results. And for future research, we can test more caffeine levels, do more trials, ensure long-term effects like growth and survival. We can also see how caffeine affects behavior after hatching. Our results support our hypothesis.